So from what I'm getting from you guys on Blackboard and your questions, it seems like immunity's kind of tripping you guys up a little bit, which is completely understandable. It's a really complex concept. And how you kind of go through immunity presentation, there seems to be some questions about that. So I wanted to take a few minutes and put together this sort of whiteboard slideshow for you guys to talk a little bit more about immunity and maybe help it make sense for those of you guys that need it kind of drawn out and put together in a more visual form. So first thing, immunity. The main goal, immunity, it wants to keep the shady things out. That's going to be our overall goal of this immunity. And if they get in, how do we deal with them? So that is where we are going to start. So first, what are these shady things? The shady things are the pathogens, the things we do not want in our body. Think proteins, viruses, bacteria, parasites, fungi. All the things we're going to talk about in more detail in this course. The immune system's job is to keep those things out. So the first thing your immune system will do in order to try to keep them out, that's its goal, is first thing it comes across is our barriers and our first line of defense. These are some of the things that you're born with. Your skin, oils in your skin, pHs of different body systems, especially the stomach acid, mucous membranes. Gravity simply works, trying to pull things down and through our system. And normal flora that exists to give some competition to these pathogens if they get in and help us not let these pathogens get any farther. All right, so then what happens if the barriers don't work? They get through that first line of defense. They're in. Secondary goal now is let's kill them. So we've moved on to our second line of defense. Things that are going to fall into here are things like fevers, complement cascades, alternate pathways, interferons. Those are crazy big words I just threw at you guys. I don't expect you guys to fully understand all of those aspects, but I want you guys to at least have heard the words because they do factor into our immune systems. The main type of second defense that we're going to focus on is inflammation. All right, so inflammation. We talk about the cardinal signs of inflammation. These are things you guys are going to want to commit to your memory. Red, hot, swollen, pain, loss of function. Those are our cardinal signs of inflammation. When inflammation is occurring inside our body, the white blood cells are rushing to the area. And they're rushing to the area to really bring the weapons to the fight and figure out how are we going to kill these guys. This is going to be the first cells that see these true pathogens and say, how are we going to get on top of this and overcome these pathogens? And on the next slide, we're going to talk more about phagocytosis, and that's really the first stage in how we start to kill these pathogens once they get into the body. So these are the first three stages of phagocytosis I've drawn out for you guys. First one is chemotaxis, where that pathogen has entered the body and the white blood cells are just starting to sense it. So this is where those white blood cells are rushing to the area. Now the next stage in phagocytosis is attachment, and this is where that pathogen actually attaches to one of these white blood cells. And in the third phase of phagocytosis, that white blood cell, once it senses that there's a pathogen attached, is going to start engulfing it. So our next stages of phagocytosis is when that pathogen is now going to become inside this white blood cell. A phagosome is going to form, and a bunch of lysozymes are going to start running to this phagosome to start digesting the pathogen that's inside the cell. So you can see digestion's occurring. At the end of digestion, that phagosome is just going to have a broken up whole chunks of pathogen in it. So pathogen can't really do anything anymore. It's been digested, it's been broken up. And then the next cool thing that's going to happen that we're going to talk about in my next drawing here, but those little broken up pieces of the pathogen are going to be inserted into part of a cell. And that's going to be our next important stage of phagocytosis. So the last stage of phagocytosis is where this antigen from that pathogen has built itself into part of the cell and is now on the outside, and the cell has become what we call an antigen presenting cell. You'll see it abbreviated as APC. So this white blood cell is now presenting the antigen, and it's basically saying, hey guys, look what I caught. Do you have something specific to help? So it is presenting this antigen to the rest of the immune system and saying, I got it. What do you guys have to help me? Because this is what our fight is up against. So something I probably should have said before we grabbed into phagocytosis, but the cell that is doing the phagocytosis is actually called a phagocyte. It is a type of neutrophil that we call a macrophage, and its main job is to eat things. So the next ones we're going to be talking about is we're going to be sort of transferring from this nonspecific innate immune system to the specific immune system. So this is kind of where we left off, right? We just talked for the last few minutes about the nonspecific immune system, innate. This is the stuff you're born with. The first line of defenses, those are your skin, your mucous membranes. And then we just talked about inflammation and phagocytosis, which is the second line of defense. So essentially your nonspecific and your innate immune system is saying to you, I don't know who you are, pathogen, but if I can't keep you out, I'm going to eat you and get some inflammation going to get me some help. And it's going to be getting that help by creating those antigen presenting cells. So then once we move into the specific and acquired immunity, this is going to be the type of immunity that's going to be based on exposure. So from here, you can look at our sort of main actor cells. When we're dealing with the specific immune system, the main actor we're going to be dealing with is lymphocytes. Now remember back with the innate immune system, we were talking about neutrophils and those macrophages, which are a type of monocyte. Those were the main actors in the innate immune system. When it becomes specific, we're looking at lymphocytes. There's going to be two different types of lymphocytes we talk about, B cells and T cells. So there's B lymphocytes and T lymphocytes. For now, we're just going to talk about the B cell one. The B cells, when they get involved, they're going to be attacking pathogens that are free-floating. So pathogens that are extracellular. So body cells, and you see the pathogens, they're floating around outside of them, but they have not gone inside. This is going to be something we term the humoral response. So B lymphocytes involve the humoral response, extracellular pathogens. And then when we add in the T lymphocyte or the T cell line, there's actually two different types. There's some T helper cells and some cytotoxic T cells. That's not too important for you guys, but just know that there's the T cells that are going to be helped with cell-mediated responses. Then these are going to take over once those pathogens have gotten inside of the body cells. So when they're intercellular, now we need to turn to our T lymphocytes to see what kind of help they can provide for us to fight off these pathogens. So when we move into the humoral response, remember this is going to be run by B cells and lymphocytes. We're only going to be looking at things that are extracellular, and it's going to be specific. So you can see I've drawn some B cells for you guys here, and these B cells are going to have on them an antibody. We also call this immunoglobulins, which is going to be abbreviated IG. That'll become important when we start talking about the different types of immunoglobulins. You guys may have already heard of some of these, like IgA, IgM, IgE. Those are all different types of immunoglobulins. Now these B cells are going to be specific for a pathogen. So you're going to have B cells for COVID. You're going to have other B cells for the flu you got last year. You're going to have B cells for every vaccine you've ever gotten. They're going to be living in your body. So remember here we're dealing with the extracellular pathogens. So you can see I've kind of drawn out some cells for you and a couple different types of pathogens. We have some green squares and some yellow triangles and some B cells that have antibodies for the yellow or green pathogens. So this is what's happening in the body. And remember back with that innate system, we had that antigen presenting cell that caught a bad guy and it presented it outside and it said, hey, I caught this. Can somebody come help me? Well, this is where it gets super cool because here, let's say we can see here that it caught, right, this like orange antigen. And right over here we have a B cell that's orange and ready. So this little B cell here is going to say, I can totally help. I have an antibody for that antigen that you caught. Come in, I'm here to help you out. Okay, and I've tried to simplify this down to just kind of the essential two things that can happen at this point. The innate immune system called out for some help. It presented the antigen. The B cell said, I got this. I matched that antigen. I have an antibody for it. They came over and they attached. And now this B cell kind of has two fates. It can, number one, it can clone itself. So it can say, all right, we have this issue. I have the antibodies to knock it out. And it makes several thousands of copies of itself. These are what you've probably heard of called memory cells. And they're what hang around for years. So when we're talking about vaccination, we want memory cells. The goal here is that the next time your body sees the pathogen, there's more B cells that are specific to it. So the response is going to happen quicker they're going to get to the cells that need help faster, and they're going to shut down the infection faster. So if they've been vaccinated for distemper, they're going to have memory B cells for distemper. If the animal gets infected with it, those memory B cells are going to rush to the site and say, nope, not going to happen. These are also what helps speed up our amnestic response, or the secondary responses. The second, third, fourth time we see the pathogen, the quicker our body's immune response is going to be for it. So number one, the B cell can clone itself. This B cell can also become an antibody factory, and both these things can happen. When it becomes an antibody factory, this B cell is just taking all those antibodies inside and it says, oh my gosh, we need these antibodies and we need them right now. So within itself, it just produces 2,000 antibodies per second, every plasma cell of these antibodies, and then it just shoves them out into our bloodstreams. So these become free-floating antibodies that are just floating around, hoping that they run into the specific pathogen. But at 2,000 antibodies a second, we can be producing a lot of antibodies just to throw out, and when they come across the right pathogen, they attack it and they take it out. So humoral response, extracellular, specific immunity. Remember, these are only going to be specific. So if this is a vaccine for distemper, these B cells are only going to work for distemper. They're not going to work if the animal is exposed to rabies. They are specific for the particular pathogen. So when we get to the cell-mediated response, remember this is still a specific immune response, but this is when the pathogen has gone intracellular, so it's infiltrated into those body cells. We're going to talk about two types of T cells. First we're going to talk about is the T helper cell. And I've simplified this down so much. It's a very, very complicated process. You could spend years studying T cells. But the T helper cell is basically there to help out the B cells. And it does a double check of the antigen presenting cell. So this is supposed to help out so we don't have any autoimmune issues. But as you start talking about diseases, things can go wrong in the immune system that can lead to disease. So the T helper cell is supposed to double check that B cell and say, yeah, I agree, it's a bad guy. You get going, clone yourself, make the antibodies. We got to get on the ball. So then the next type of T cell is the cytotoxic T cell. And this is really a direct cell killer. So what we have happening, you can see I've kind of drawn out that body cell, right? And it's infected. It's got a pathogen inside of it. So this body cell is going to sort of present that antigen outside of it and say, hey, I'm infected by this pathogen. It's not good. And it's trying to take over. So the cytotoxic T cell will see that, and it will attach to that body cell. And once it attaches, it sends a signal inside that cell. It says, yep, you are right. You are infected. I'm sorry, but you need to die. And it sends a signal in so that that cell can die, and therefore the pathogen cannot proliferate outside of that body cell. So this last slide is just a flow chart for you guys if you're asking what type of immunity. So this is helping us figure out if things are active, natural immunity, artificially active, natural and passive, or artificial and passive. So take a look at it. If you were confused by this, hopefully the flow chart will help you figure out which type of immunity you're looking at.